Welcome back, guys. We are having a new video this time for the biomolecule series. Almost second last video, I think. Then it will be completed. I'm trying to finish it as soon as possible. In that case, in the last video, we have completed the protein. Okay. So this time, I'm going to complete nucleic acid. Okay. So be with me. This side Govind and this is our chemistry channel, Chemistry Guru 1993. Thank you, children, for supporting. So check it once what we are going to discuss today. What is the today's topic? Today's topic is nucleic acid. This is the today's topic, nucleic acid. So what is nucleic acid, children? Actually, again, I'm not very good in drawing, but I'm trying. Here we are having cell. Okay, so this is our human cell, and in the cell here these points, these are the nucleus. Okay, and these nucleus are formed by chromosome. So chromosome just like this. Okay, further they will be divided into like this. It's a DNA structure. So it's a cell, human cell. In that cell, nucleus is there, and that nucleus is made by chromosome. This is your chromosome, and these chromosomes are made by DNA and RNA. Okay, DNA and RNA. So basically nucleic acid made by DNA, RNA. Full form of DNA and RNA: deoxyribonucleic acid. Deoxyribonucleic acid. And RNA, ribonucleic acid. Ribonucleic acid. This is the full form of DNA and RNA. So it's a kind of a small information. So basically, we'll talk about the nucleic acid this time. In the nucleic acid, children, what we are having in the nucleic acid basically, so listen it once carefully. Nucleic acid, in the nucleic acid, children, nucleic acid is formed by basically from three things. What are the three components? One is pentose sugar. Pentose sugar. Second is phosphoric acid. Phosphoric acid derivative. And the third particle is nitrogenous base. nitrogenous base. So basically these are the three substances from which your nucleic acid form: pentose sugar, phosphoric acid derivative. In the pentose sugar we will use ribose and deoxyribose. Ribose and deoxyribose, that will be we will use, and both ribose and deoxyribose beta form. What? Beta form of ribose and deoxyribose. Here in the case of phosphoric acid derivative, phosphate ester, phosphate ester we will use. In the case of nitrogenous base that we will discuss, basically they are heterocyclic. Heterocyclic. Okay. So beta form of pentose sugar we will use, phosphoric acid derivative, that is phosphate ester, and nitrogenous base. These are the things what we are going to use in the case of formation of nucleic acid. So let's check it one by one. First one is ribose and deoxyribose. Ribose and deoxyribose. So what is ribose? So what I said, it's a pentose sugar, that means 5 carbon. So first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. On the first carbon, aldehyde group is attached. Second carbon H, OH, here OH, here OH, last CH2, OH, remaining hydrogen. Okay, so this is your beta ribose. What it is? Beta ribose. Basically it is a ribose. Now check the beta and alpha. In that case, this oxygen lone pair, same thing, it will approach carbon, these electrons shifted, and this hydrogen will attach here. So what we are getting now? Here we are getting, now directly the cyclic structure I am going to form for the beta. State chain structure if you want to check, so it is like that: 1 2 3 4 and 5, CH2OH, here OH, and here H, and here OH. Same work here and transferred. So what is the cyclic structure, children? So here it's a 5-membered ring. Carbon number 1, H, OH, then OH, H, here also OH, H, and here CH2OH, and here H. This is the carbon number 1, carbon number 2, 3, and this is the carbon number 4. So it's your beta ribose. So experimentally it is found that in the nucleic acid beta ribose is used. for for deoxyribose, that is also very simple. Same time, what you have to do, children, you have to remove oxygen from the carbon number 2. what? C2 from the carbon number 2 remove oxygen. From C2 remove oxygen, then you will get deoxyribose. So what is the cyclic structure for deoxyribose? From this carbon you have to remove oxygen, that's it. So here, as it is, OH, H, here H, here H, here OH, here H, here H, and here CH2OH. So from this carbon only I removed oxygen. So it is beta deoxyribose. So these are the pentose sugar, ribose and beta D ribose. I hope it is clear to you. So they are the derivative of furan because five membered ring. So in nucleic acid only beta form is exist. That's why beta D. So you can say beta D ribofuranose, derivative of ribose. So ribofuranose. Similarly, this beta D ribofuranose. These are the also name. Now come to the, this is about the ribose and deoxyribose children. Once again I am telling you, this is the ribose cyclic structure. From the ribose carbon number 2, if you are removing oxygen, then you get deoxyribose. That's it. Second, number 2, phosphoester. Phosphoester. In the case of phosphoester children, what is phosphoric acid structure? Phosphorus, double bond, oxygen, here OH, one more OH, one more OH. So this is the structure of phosphoric acid. In that case, if you are removing hydrogen and adding R, okay, then it is converted into OR. Okay, so that is your phosphoester. Mono, if you are removing one H, then mono. If you are removing two, then di. Removing three, then tri. Okay, basically diphosphoester we will use. So nucleic acid contains only monophosphoester, sorry, diphosphoester. So in that case, diphosphoester, that means here phosphorus, here minus, here minus, and here OH. This is your diphosphoester. Okay, that we will use. So that is the phosphoester. Now come to the third, nitrogenous base. What is nitrogenous base? Nitrogenous base, total 5 type of nitrogenous base we are having, and they are the derivative derivative of purine and pyrimidine. Pyrimidine. Okay, what I am saying, they are the derivative of purine and pyrimidine. Okay, so nitrogenous base derivative of purine. What is the structure of purine, children? Check it once here. See, check it properly because it is not easy and no need to learn. Okay, because it is not required, but for the information you have to know. That's why I am writing this here. One nitrogen atom here, one nitrogen atom, nitrogen, and here nitrogen, and here double bond. So number 1, 2, 3, 4, 5, 6, 7, 8, and 9. This is the purine. What it is? It's a purine structure, children. Okay. What about pyrimidine? This is the structure of pyrimidine. Here one nitrogen. Wait. One nitrogen here and one nitrogen here, and alternate double bond. 1, 2, 3, 4, 5, 6. Okay. So these are the structure of purine and pyrimidine. From the purine and pyrimidine, we'll get nitrogenous base derivative. They are the derivative, not purine and pyrimidine. They are not nitrogenous base. From there we can derive. Okay. So check it once. From the purine, I hope this is noted. From the purine, this is the purine, guys. From the purine, two nitrogenous base will be generated. What are these base? At the carbon number 6, you have to add NH3, NH2. Okay, here nitrogen, one more nitrogen here, one nitrogen here, and one nitrogen here, and double bond. This is the double bond condition. Here you have to add NH2. Okay, this this is called adenine, ADENINE. That is the base derived from the purine. Okay, next Guanine. How guanine will be generated? Check it once. Here, for the guanine, I am telling you, this is the structure of guanine. Here one nitrogen here, nitrogen, and here nitrogen. Double bond, double bond, double bond, double bond. This place NH2 will be attached. This nitrogen, hydrogen is attached. Here double bond is attached, remaining as it is. It's called guanine. This is the second nitrogenous base. First nitrogenous base adenine and guanine. These are the two nitrogenous bases which are derived from the purine. Now come to the pyrimidine. Okay, what is pyrimidine, children? Pyrimidine structure is this one. One nitrogen here, one nitrogen here. Okay, it is your pyrimidine. Three nitrogenous base will be generated from here. What are these three? Number one nitrogen, second nitrogen, double bond, and here NH2. With this nitrogen, one hydrogen is attached. It's called cytosine. One nitrogen here, one nitrogen here, double bond. Oh, ho, 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 one double bond missed, only one double bond. Here, the only one double bond is present, remaining one nitrogen here, one nitrogen here, hydrogen is attached. In this place, oxygen with double bond, one hydrogen is attached with this nitrogen, and one oxygen with this uracil. This is the third one, means second one in the case of pyrimidine. Now, third and last nitrogenous base. Here CH3 is attached. Here nitrogen with hydrogen. Here double bond oxygen. Here hydrogen, and here that is thymine. So all the five nitrogenous bases we have completed, and the formation of nucleic acid will be done by these three things. What are the three things? Pentose sugar plus nitrogenous base plus phosphoester. So this is the combination for the amino acid. So remaining discuss nucleoside, nucleotide, we will do in the next lecture. This is all for today. Thank you guys, and all the best for your preparation.